Today we going to discuss the chapter number 3, and you should be able to describe the structure and function of the cell membrane, including its regulation of material into and out of the cell. Describe the functions of the various cytoplasmic organelles. Explain the structure and contents of the nucleus, as well as the process of DNA replication. Explain the process by which a cell builds proteins using the DNA code. List the stages of the cell cycle in order, including the steps of cell division in somatic cells. Discuss how a cell differentiates and becomes more specialized. List the morphological and physiological characteristics of some representative cell type in the human body. You develop, as a human being, you develop from a single fertilized egg cell, sperm from the father and the ovum or the egg from the mother. The cell first divided into millions of cells. These cells then differentiate to become specialized in their structure and function. Differentiated cells then form tissues that work together to perform particular function and contribute to homeostatic or homeostasis, dynamic state of balance within parameters. And we spoke about homeostasis last week. So to keep your body within like the normal range, physiological range. And we'll give about positive feedback, negative feedback, etc. So if you still don't know them, please review them. 2 or more tissues from organs, and organs from organ system. Why study cellular level of organization? Basically, this makes you understand the physiology of the body. Cellular and developmental biologists study how the continued division of a single cell leads to such complexity and differentiation, and structure of different cells and how the structure correlates with the function. For example, take a look at the skin cell here on the left side versus nerve cell. They differ in structure and function. The cellular components, as they are responsible for the thousands of biochemical reactions necessary for an organism to grow and survive. Despite differences in structure and function, all living cells have surrounding cell membrane that they have to interact with other cells. They have to interact with environment. So what is the structure and composition of the cell membrane? It's very important because, as you know, cell membrane actually protect the cell and regulate what can get in and what can get out, etc. So has very specific plasma membrane. It's made up of phospholipids, cholesterol, and various proteins. A single phospholipid molecule is made up of phosphate group on one end, called the head, and the phosphate group is negatively charged, making the head polar and hydrophilic. Hydrophilic, polar, charged, hydrophilic, loving water. So L from love, hydrophilic. 2 side by side chains of fatty acids that make up the lipid tail. The fatty acids are uncharged, making the tail nonpolar and hydrophobic. Phobic from phobia. They hate water. They don't like water. Hydro, water. Phobic, they don't like water. Fatty acids can either be saturated or unsaturated. We're going to see that there is one leg saturated, the other leg is unsaturated. Phospholipids are thus amphipathic molecule. But that means it contains both hydrophilic and hydrophobic regions. Okay? This is actually the phospholipid structure. You have the head here, where you have the phosphate, and you have the glycerol, and then the tail here has saturated fatty acid and unsaturated fatty acid here. So this is the hydrophobic from phobia, they don't like water, and the head actually like water. That's why the tail always hidden inside. So this one facing extracellular and intracellular membrane. Those in the middle hidden away from the water. The cell membrane consists of two adjacent layers of phospholipids called phospholipid bilayer. By two. So it has, as you can see here in the image, in the bottom, you have two phospholipid bilayer. Bilayer means two layers. So this is one layer, this is another layer. This is facing extracellular, where you have the head. The head loves water, that's why it can get along with water. And the intracellular as well. Those heads can get along with intracellular content here of water inside the cell. Lipid tail face each other, whereas the phospholipid heads face outward. Phosphate group of one layer interact with intracellular fluid, and phosphate group of the second layer interact with extracellular fluid, as you can see here. The lipid bilayer is also composed of two types of proteins. You have integral protein are embedded within the membrane. So when you hear integral protein, it's actually within the membrane. It's like embedded inside the membrane itself. And it has actually channel protein, selectively allow particular materials to pass in and out of the cell. So I have channel protein that allow some material to get in and out of the cell, and the receptor protein is a type of recognition protein that can selectively bind a specific molecule or ligand. So these guys is very specific, so they allow a specific molecule to get in. Channel protein can a specific particular material to get in and out. The glycoprotein is a protein that has carbohydrate molecule attached, extending into the extracellular matrix. The carbohydrate attached into the protein aids in cell recognition. So this is like a flag. You know, when you raise flag just to show the building, you have the United States flag on the top of the building, stuff like that. So it's actually, this is the carbohydrate attached into the protein aids in cellular recognition. So specific molecule can come and bind to it. And you have the peripheral proteins are typically found in the inner or outer surface of the lipid bilayer. They have various functions such as digestive enzyme. See the beautiful bilayer of the cell membrane? This is the glycoprotein right here, protein with carbohydrate attached, same thing here. And this is the channel protein we talked about. This is some cholesterol. This cholesterol actually make the cell membrane mosaic, I mean flexible a little bit, to give it some flexibility. And this is the integral membrane protein embedded within the cell membrane, as you can see. And this is peripheral membrane protein. And this layer here, this is the phospholipid bilayer we spoke about. So what is the function of cell membrane? You know, cell membrane to protect the cell and to regulate what can get in and out, so no harmful material can get in, or no valued material or valuable material can get out. So the structure of plasma membrane causes the membrane to be selectively permeable, allowing only relatively small nonpolar substances to move through the lipid bilayer. Example: lipids, oxygen, carbon dioxide, and alcohol. In case of large and polar substance, need some assistant. These need some assistant to cross the membrane because they are repelled by hydrophobic tails of the phospholipid bilayer. Example be the glucose, electrolytes, and amino acid. How do substances move through the membrane? There are two types of transports through which substance move in and out of the cell across selectively permeable membrane. You have passive transport and active transport. Passive transport will be easy, as you know, we explained in the lab before, that if you have, for example, if you have, let's say you have, this is a ball, okay, and you have like 5 steps. Okay, if you have a basketball or soccer ball in the top here, with a little bit of wind, this ball can go down, right? This is exactly what happened in the passive transport. It's easy. You go from high concentration to low concentration. But in the opposite situation, when you have to kick the ball high from the first steps to the 6 steps, for example, you need energy. This is what we call active transport, and they're gonna be ATP involved. Okay. Passive transport allow movement of substances down the concentration gradient without use of ATP. And as we explained before, ATP, the energy that your body use, it's like when you drive the car cannot move if you don't fill it with the gas. So this is your biological gas. Without it, you cannot even blink an eye. We spoke about it before. And the substances move from an area of higher concentration to low concentration is easy, right? So you don't need energy. But there are 3 types of passive transport: simple diffusion, facilitated diffusion, and osmosis. This is a cell membrane, and this is the bilayer here, the phospholipid bilayer. And you have, this is an example of the simple diffusion. Okay, by looking just the structure of the lipid bilayer, allow only small non-polar substances such as oxygen and carbon dioxide to pass through the cell membrane down their concentration gradient by simple diffusion. So if this is like extracellular, this is intracellular. These non-polar molecules have a lot outside, as you can see, and a little bit inside. So by the time pass by, those molecules start to get in with the simple diffusion inside the cell here. See that, compare this from this one, from this one, from all these molecules now by passing the time. Okay? Facilitated diffusion, like you need someone to facilitate the entrance of the molecule. So facilitate diffusion of the substance crossing the cell plasma membrane takes place with the help of proteins such as channel protein, okay, and carrier protein. Channel proteins are less selective than carrier protein and usually mildly discriminate between their cargo based on size and charge. Okay, the carrier protein are more selective, often only allowing one particular type of molecule to cross. Okay, and the osmosis, as you can see here in these 2 jars, which compartment has more water now? The left side, right, have more water. So it's the movement of the water of the water from high concentration to low concentration. So this on the right side has like hypertonic solution, which has a lot of solutes but less water. This on the left side here has a lot of water. So water will be moved from this compartment, higher concentration, to the low concentration. You see, through the semi-permeable membrane. So here, after passing the time, a lot of water went to the other compartment here, from high concentration to low concentration. This is just to show you the type of solution we talked about before. Hypertonic, mean a lot of solutes, less water. Isotonic, equal. Hypotonic, have a lot of water, more than the solutes. And we spoke about the blood as example. cell membrane. Now, example of the sodium-potassium pump. This actually what keep the, especially in the nervous system, in the neuron, keep balance between the sodium and the potassium. As you know, the electrical charge always they're gonna drain. Like if you when the action potential happen, a lot of sodium rushing inside the cell. If you don't replenish the outside with the sodium will degrade it. That's it. There's not enough sodium to outside the cell to create action potential or electricity. Same thing, by the same token, there is not enough potassium inside the cell to create another action potential. So the sodium usually outside is higher. So when there is action potential, a lot of them rushing in, and some potassium rushing out. So if you don't balance this situation using the sodium potassium pump, there is no action potential or electricity activity will happen again in the cell. So the sodium potassium pump is found in many cells, many cell plasma membrane, powered by the ATP. The pump moves sodium and potassium ions in opposite direction, each against the concentration gradient in a single cycle of the pump. It's exactly like moving 3 sodium from inside. After the action potential, you send them outside, but same one is gonna bring 2 potassium inside to keep the balance of this cell. This is example of the phagocytosis, against large particles, pinocytosis, small particles. Does the same job. See, it's engulf around it and make vacuole, which is a little bigger than the vesicles. The smaller pinocytosis, it has small vesicles. Now the receptors mediated endocytosis, you see that this receptors is like lock and key. So this ligand will bind to the lock. Once it binded, it's gonna get it inside the cell, endocytosis. This is coded vesicles, okay? Exocytosis, as we explained, is much like endocytosis in reverse. Material destined for export. So exit or export. So we want to send something outside the cell. We spoke about the cytoplasm and cellular organelles before, but we're gonna touch base a little bit about it too, because I think we had it in the lab, but for those who didn't attend the lab, we just gotta review it. Cell membrane surround the cell, separating external environment from the cell's cytoplasm. And of course, we spoke about the cell membrane before, and we know it's phospholipid bilayer to protect the cell, right, and to control what's get in and out. So it's like bilayer phospholipid with the heads facing outside the cell and inside the cell, and the hydrophobic tail will be in the middle, cell membrane. Cytoplasm comprises of cytosol and organelles, and the cytosol itself is the jelly-like substance within the cells, providing the fluid necessary for biochemical reaction. So a lot of fluid inside the cell. And the organelles, what we mean by organelle, it's a like membrane enclosed body in the cell within a unique function. For example, you have the mitochondria, it's organelles, nucleus, organelles, Golgi, organelles, rough endoplasmic reticulum, organelles. Anything bound or closed called organelles. The cytoplasm and cellular organelles. So endoplasmic reticulum, as you know, we spoke about it before. This is like the kitchen that actually manufacture the protein. And along those huge kitchen, you have this like experienced chef called ribosome that connect the protein together or make the specific protein that the cell need. So that the endoplasmic reticulum is winding network of thin membranous sacs found in close association with the cell nucleus. It has to be very close to the cell nucleus because it receives the order from the cell nucleus. Rough endoplasmic reticulum is studded with numerous ribosomes, which are sites of protein synthesis, because you know the ribosome actually the experienced chef that actually can cook the protein. And the smooth ER synthesize phospholipids, steroid hormones, regulate the concentration of cellular calcium, metabolize some carbohydrates, and break down certain toxins. Lysosome, actually this is one of the defense mechanisms that the cell defends itself against any harmful material. Lysosome is an organelle that contains digestive enzymes that break down and digest unneeded cellular components such as damaged organelles. The mitochondria, the powerhouse, very important, and so this is where you make the ATP inside this. And a mitochondria is composed of two separate lipid bilayer membranes, an outer membrane and inner membrane. The inner membrane is highly folded into structures called cristae, along which various protein, enzymes, and other molecules perform cellular respiration, responsible for the ATP synthesis or the production of the ATP. So inside here, you make a lot of ATP. Peroxisome, as you can see lipid bilayer here, membrane-bound organelle that contain abundance of enzymes for detoxifying harmful substances and lipid metabolism. Peroxisomes produce large amount of toxic H2O2, hydrogen peroxide, in the process of detoxification, but they enzymes that convert the hydrogen peroxides into water and oxygen. The cytoplasm and the cellular organelles continue. We have three components of cytoskeleton. As we explained, cytoskeleton give actually the shape of the cell and protect the cell against like pressure and elongation, etc. So you have three types: microtubules, which is the thickest one of them, a thick cytoskeletal filament composed of tubulin subunit. Microtubules maintain cell shape and structure and help resist compression of the cell, play a role in positioning the organelles within the cells, and assist separation of DNA during cell division. They are also part of the cilia and flagella. The microfilaments is the thinnest cytoskeletal filament, forming twisted chain mostly made up of actin protein together with protein myosin and responsible for muscle contraction. We're gonna see this in detail when we spoke about muscle contraction. Is the bridging between thick and thin filament or thick and thin protein called actin and myosin. The intermediate filament is intermediate in thickness, composed of long fibrous subunit of keratin protein that are wound together like the threads that compose a rope. Together with microtubules, intermediate filaments are important for maintaining cell shape and structure. The nucleus. We spoke about the nucleus, and we know that this is the brain of the cell. We have all the secret ingredient to make any protein. So the nucleus is made of double membrane called the nuclear envelope. Spanning these two bilayer are nuclear pores, which are tiny opening for the passage of proteins, RNA, and solutes between the nucleus and cytoplasm. The nucleus contains the genetic material in form of DNA, deoxyribonucleic acid, that determines the entire structure and function of the cell. The nucleolus, the smallest one, the smallest version of that, the nucleolus, right, is located inside the nucleus and responsible for manufacturing the RNA, which then aids in ribosome synthesis. DNA macrostructures. Strands of DNA are wrapped around histone protein. This is a histone protein at the top here. We just use the pen. So this is a histone protein here, forming nucleosomes. DNA with histones is packed tightly into chromosomes. When the cell is ready to divide, before cell division, when DNA must replicate chromosome loosens up to form chromatin. This one. So it's packed, and then you want to stretch it to start make copy of the DNA, as you can see here. So take from the chromosome, stretch it a little bit, and after that's gonna unwind it to read a specific sequence or to make copy of the strand. So the molecular structure of DNA, the DNA double helix is composed of two complementary strands. As you can see here, these two are complementary with each other. The two strands are bonded together via their nitrogenous base pairs using hydrogen bond. It's very important that we're going to see that the nitrogenous base is very important. The nitrogen base is made up of pentose sugar, pentose 5, right, and phosphate group, and nitrogen base. And you have only 4 nitrogen base, A, T, C, G, and we're gonna speak about it that A always go with the T on the other side and vice versa, and C always go with the G on the other side and vice versa. Okay, the DNA replication, and in order for an organism to grow and maintain its health, cells must reproduce themselves by dividing to produce 2 new daughter cells, each with full complement of the DNA as found in the original cell. DNA replication always occurs before cell division. Definitely, because let's say this is one cell and has a nucleus and the cell membrane and other organelles, right? These secret ingredient here, or the textbook of cooking, and as we give the example last time, this is secret ingredient. You need actually to make 2 cells. You have to make copy of this secret ingredient here. So instead of 1 textbook, you have to have 2 exact textbook to make 2 cells. How actually the DNA replication? It happened actually in stages. First stage called initiation, second stage called elongation, and third stage called termination. In the initiation, 2 complementary strands of DNA separate with help of a special enzyme, including helicase. So you have to separate them first, okay? And then in the second elongation stage, each strand becomes a template along which a new complementary strand is built. An enzyme called DNA polymerase help elongate the new DNA. So separation, initiation, and then elongation. Okay, third one will be the termination, where actually each strand of this will add the complementary other half of it. So you have actually 2 DNA. Termination, once the 2 original strands are bound to new complementary strands, DNA replication is stopped and 2 new identical DNA molecules are completing. The DNA replication is called semi-conservative because each new DNA molecule contains only 1 strand from the original molecules and 1 newly synthesized strand. So this is the original. You elongate them, they initiate, and you elongate them, and then you added whatever the second strand here, new second strand here. So each one from the 2 DNA carry 1 of the old one and 1 new one, 1 old one and 1 new one. The protein synthesis is a little bit like close to the process of DNA replication, but this time would be like into codes called coding. So the DNA holds all of generic information necessary to build cells protein. The nucleotide sequence of a gene holds recipe for particular type of protein. The sequence is read in triplicate called codons. Each codon codes for a specific type of amino acid, which building block of every protein. So for example, see this two-letter AUG for this particular amino acid. This 3 CCG for proline, like a certain amino acid. CAA for GIN amino acid. UCU for serine amino acid. GUU for valine amino acid, and so forth as you go. So each actually amino acid, the code has 3 nitrogen space, okay? But as you see here, if this is a template, you see this is a template, a double helix. So let's say this piece, okay, let's say I have like 46 chromosome, right? You have 46 chromosome, and this protein, actually how to make this specific protein, the information located on chromosome number 10, for example. So this is a chromosome number 10, double helix, okay? So specific RNA is going to come and just unwind it, this piece, this is specific piece here, for example, unwind it and read the sequence of the nitrogen space and take it to nucleus. When they take it to the nucleus, nucleus is gonna add another to the other side, whatever base is missing, and make this specific protein. How they make it? With every 3 codon of this. Here T, on the other side A, here A, other side T, C goes with G, G goes C, etc. 4,000, even million of sequence. But when the replication of the RNA in the transcription, there is no T. We replace the T with U. Remember this. So replace T with U. So instead of T, there is U. Whenever we see U here in the transcription, originally in the DNA it was T. Okay? This is the transcription from DNA to messenger RNA. And the first step of making protein from DNA, nucleotide sequence in a specific gene is transcribed or copied into complementary messenger RNA. Okay, so this is a transcript, this is the RNA polymerase. Okay, here this is the template strand again, and we take this copy here, transcription, to make this copy, and then we're gonna make the other strand. Ribosome is going to put other strand. A is going to put next to A U, A, G, C, C, G, etc. So transcription begins in a fashion somewhat like DNA replication, but only a small specific region of DNA where the gene is located unwind. Initiation, we spoke about initiation, a region at the beginning of the gene called promoter is where the RNA polymerase begin transcriptions. Elongation, RNA polymerase reads nucleotide sequence on the gene and transcribes the information into nucleotide sequence in the messenger RNA. The only difference is that messenger RNA molecule does not contain T. Remember, instead it has U. Okay? Stage 3 termination, when the RNA polymerase has reached the end of the gene, one of 3 specific triplets, this is like a stop sign. It's UAA, UAG, UGA. Yoga. Remember yoga? UAA, all start with the U, double A, AG, GA. Cause a stop signal which triggers the enzyme to terminate transcription. Now the translation, okay, we're gonna translate from RNA to protein. Translation happen with every 3 nitrogenous base hold code for 1 amino acid. You know, protein made of like a lot of amino acid, but it has to be in a specific sequence. Okay, so the name of the amino acid you can break down, if this is amino acid, for example, it has to have 3 letters, the name. This is called the code for it, okay? So during translation, the messenger RNA transcripts is read by ribosome and transitional RNA molecules. As ribosome slides along the sequence of messenger RNA, transitional RNAs bring the appropriate amino acid in sequence to the growing polypeptide chain by matching their anticodons with codons on the messenger RNA. Okay, so this is like 3 letters, 3 nitrogenous base here, AUG. This is the abbreviation or the secret name or the code name of a specific peptide. Okay, so here, as you can see, it's AUG, right? What do you need with the AUG? On the other side, A need U, U need A, G need C, right? So here, as you can see, UAC, AUG, on the other side, ribosome bringing the other complementary will be UAC. A has to have U on the other side, U have to have A on the other side, G have to have C on the other side, and so forth. Start to building those peptide until you reach a lot of polypeptide. RNA is read in triplicates called codon. Okay, each codon represents particular type of amino acid. As you can see, these 3 letters is coding for a specific amino acid. Another CCG for proline, for example, amino acid, and UCU for like serine, GUU valine, and so forth. So this is the name of the amino acid. H3 letter is the specific code for specific amino acid, and when you put like specific sequence of amino acid with each other, it's going to make specific protein. Now the cell cycle, we spoke about it in the lab as well. We're gonna just to review it again. As you can see, the cell spends 75% of each life span preparing for the actual mitotic phase here. So G1, growing, preparing for the synthesis, and have the enough enzyme and enough material to be able to go from one cell to 2 cells here. So have two main part of the cell cycle: interphase, which is the green here, the gray one, the huge one, the big one, 75%, and the mitotic phase, the actually split or the copy of the one, we call it mitosis. Interphase is the period of time when cell is not dividing. It is divided into following phases. G1 phase, cell grow in size and carries out all normal metabolic function. S phase, DNA synthesis, replication occur. G2 phase, cell continues to grow and makes necessary preparations for mitosis, and the G0 is when the cell does not divide. So don't confuse yourself. You go with the G1, S, and G2. Then the actual mitotic phase. And this is the actual mitotic phase, where you have the prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. In the prophase, you have the chromosomes condensed and become visible, and spindle fibers emerge from the centromeres. Nuclear envelope breaks down. You need to break down, of course, the whole to be able to free and to be able to make copies. And in the prometaphase, the chromosomes continue to condense and the mitotic spindle microtubules attach to the Kendall cores. So start to attach to this material in the middle. Metaphase, when you actually all the copies stay in the middle, as you can see here. So metaphase in the middle, you put all the chromosomes in the middle, like if it's exactly the rooms and you want to split the rooms, you put all the student in the middle and you make 2 rows. You take one to the side and one to the right and one to the left. And the anaphase, each pool will pull the chromosomes towards, so you have the condensed split into 2, the chromosome splits into 2, and the sister chromatids, now called chromosomes, pull toward opposite poles. As you can see, they're pulling. This is pool, this is another pool. And the telophase, the chromosomes arrive at the opposite direction, as you can see here, and the number and the nuclear envelope start to form again, or start to set up the wall again, or the nuclear envelope. The mitotic spindle breaks apart, the spindle that used to hold or to attract the chromosomes toward the pool, it's break down. And finally, in the cytokinesis, you have the animal cells, a cleavage furrow, start to building a wall between the 2 cells, separate the daughter cells. In the plant cells, a cell, the precursor to new cell walls. So in the plant cell, there is cell walls. Actually, in the animal cell, there is the cell membrane. Again, this is just to show you the control of the cell cycle. The cells proceed through the cell cycle under very precise control of checkpoints, which determine whether or not the cell is prepared to move into the following stage. So those checkpoints, you always visit a checkpoint to make sure we're going from G1 to the S phase, and from the G2 to the mitotic phase, and then from in the middle of the interphase, processes to give 2 exact order. So G1 checkpoint, ensure that the cell is ready for DNA synthesis. This is on here. G2 checkpoints, ensure that the DNA synthesis occurs successfully and the cell ready to enter mitosis. I'm sorry. And the metaphase checkpoint here in the middle. You remember meta from the middle. Ensure that all sister chromatids are properly attached to their microtubules and line up in the metaphase plane. The cellular differentiation, we have a lot of cells and a lot of tissues. So how actually this happen? A complex organism such as human develop from single cell into vast array of cells type through process called cell differentiation. So differentiation, what is actually differentiation? Is a process by which unspecialized cells become specialized to carry a distinct function. Differentiation begins with the stem cell and is regulated by different genes in the genome. What is the stem cell? We all hear about the stem cell. Stem cell is an unspecialized cell that can divide without limits as needed and can differentiate into specialized cells. So actually, if you have the stem cell, if it's like a nervous system, can give you like neurons. And if you have a stem cell, if you put it in the kidney, it can give you kidney cell. If within the liver, can give you liver cell, and so forth. So this will be the last slides we discussed today, and I hope this will help. And definitely, if you have any question, we'll be able to discuss it during the class. Hope you enjoyed the lecture, and see you next week.